I'm Ain. I am undergraduate student from Department of Biomedical Science, University Putra, Malaysia. Welcome to my virtual poster session. So my study title is Sequence Analysis, 3D Structural Prediction and Epitope Mapping of Leptospira protein, Potential Diagnostic Marker for Pathogenic Leptospira. Before we go further, let's see the background of this study. Basically, pathogenic leptospira is the main causative agent for leptospirosis. So leptospirosis generally is a common genetic disease that mostly affects population in the tropical countries. However, in recent years, the current diagnostic method has posed several limitations. This happened particularly due to the increased number of survivors and the biphasic nature of leptospirosis. Thus, altogether, this urged a new discovery of potential diagnostic markers that can be found in all species and give promising solutions for the arising diagnostic issues. So, previous studies have conducted and identified that effective and reliable PCR assays are the most suitable diagnostic method, as it can identify the presence of Leptospira bacteria even in the early phase of infection. The previous study also stated LipL41 is one of the immunogenic antigens and has been suggested as one of the good candidates in developing diagnostic methods. So identification of highly conserved region of LipL41 will lead to development of reliable and accurate diagnostic kits of leptospirosis. So this study was aimed to investigate the potential of LipL41 as diagnostic marker for accurate and reliable diagnosis of leptospirosis by using bioinformatic approaches. So moving on to the method, so this study have used NCBI as the database and several other bioinformatic tools including RAS, GelView, BLAST, Muscle Alignment Method, BepiPrep 2.0, and IEDB analysis. So from this study, as we can see here, Figure 1 showed the multiple sequence alignment analysis of LipL41 nucleotide and amino acid sequences. So for 1A, highly conserved region, which indicated in the black box, was the longest conserved region from all aligned 29 pathogenic leptospira strains that identified from nucleotide position 1 to 23. Meanwhile, for 1B here, this red box indicates a longest conserved region of amino acid sequences that found from residue 42 to 74 and overall high conservation was observed. Moving on to the overlap tertiary structure of LipL41 protein, a highly overlapped structure was identified with 23% of similarity between all the structures of 7 different pathogenic leptospira species. And this table 1 showed 4 predicted LipL41 linear B-cell epitope sequences with various residual length, epitope structure, surface accessibility, and similar conservation percentage. So, from this study, LipL41 only present in pathogenic leptospira, which allow selective detection of pathogenic strains in developing diagnostic tools. Conserved LipL41 nucleotide sequence, which is seen in figure 1A, is one of the suitable target sequence for developing PCR-based diagnostic assays due to the ideal length. Then high conservation was seen in LipL41 amino acid sequence in 1B and also overlap tertiary structure have provided supplementary information on the overall function of LipL41 protein that is useful for conformational studies in the future. Then interestingly, 3 of the linear B-cell epitope predicted have shown a great potential to be used as target sequence for epitope-based diagnostics. So in conclusion, this study suggests that LipL41 nucleotide amino acid sequence and B-cell epitope has the potential to be used as early and accurate diagnostic marker to detect pathogenic leptospira. So that is all from me. Thank you.